Imagine holding a simple rock in your left hand and a living cell in your right. Between them stretches a seemingly unbridgeable chasm. One is merely matter, the other somehow alive. This transformation, from inanimate minerals to the first flickering of life, represents perhaps the greatest magic trick in cosmic history. But unlike magic, this remarkable transformation happened without any supernatural intervention. It was chemistry, elegant, persistent, and ultimately inevitable chemistry. What if I told you that scientists have identified a single molecule that might have been the bridge between these worlds? A chemical compound so fundamental to life that without it, you would not exist, nor would any plant, animal, or microbe on our planet. This is not speculation or wishful thinking, but a rigorously developed scientific theory backed by laboratory evidence. The story of this molecule takes us on a journey from volcanic ocean vents to NASA laboratories, from Darwin's warm little pond to the frozen moons of Saturn. It connects the elemental violence of the early Earth with the delicate, ordered complexity of the cell in your body right now. The question of how life began has haunted humanity since we first gained consciousness to wonder about our origins. For millennia, we had only myths and philosophical speculation. Today, we stand at the threshold of an answer that is both more extraordinary and more profound than any creation myth, because it appears to be true. Our planet formed approximately 4.5 billion years ago from the gravitational collapse of a nebular cloud, condensing into a molten ball of rock and metal. The early Earth was unrecognizable, a hell's cape of volcanic eruptions, meteorite impacts, and a toxic atmosphere devoid of oxygen. No life as we know it could possibly survive in such conditions. And yet, somehow, by 3.8 billion years ago, microscopic living cells had appeared, already equipped with the basic biochemical machinery we recognize in organisms today. In 1871, Charles Darwin speculated about life's origins in a letter to his friend Joseph Hooker. He imagined some warm little pond, with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed ready to undergo still more complex changes. This poetic vision remained just that, poetic, for decades. The scientific community lacked the tools to investigate such ancient events. The modern search for life's chemical origins began in earnest in 1953. Stanley Miller, a 23-year-old graduate student working under Nobel laureate Harold Urey at the University of Chicago, designed an apparatus to simulate Earth's primitive conditions. His setup was ingeniously simple: a closed system containing water, representing the primordial ocean, methane, ammonia, and hydrogen, the presumed early atmosphere. Miller applied electrical sparks to simulate lightning and then waited. After just a week, something remarkable happened. The clear water had turned a murky reddish-brown. When Miller analyzed this primordial soup, he found it contained amino acids, the building blocks of proteins, that had formed spontaneously from the simple starter chemicals. The implications were staggering. Life's basic components could emerge naturally from inorganic precursors. Yet amino acids, while crucial for life, are not enough. Modern cells require an intricate choreography of molecules working in precise coordination. Most importantly, they need a way to store and transmit information. This is the role of DNA and RNA, the software of life that contains the instructions for building and operating all living things. For decades, scientists faced a chicken and egg problem. Proteins, made from amino acids, are needed to build and maintain DNA, but DNA contains the instructions to make proteins. How could either exist without the other? What came first? Enter Zara Dawson, a young biochemist who in 1981 was studying catalytic properties of RNA at Stanford University. While conventional wisdom held that RNA, like DNA, was merely an information carrier, Dawson observed something unexpected. Under certain conditions, RNA molecules could fold into shapes that enabled them to catalyze chemical reactions, a function previously thought to be exclusive to proteins. This discovery revolutionized our understanding of molecular biology and offered a solution to the chicken and egg problem. If RNA could both store information like DNA and catalyze reactions like proteins, perhaps the first life used RNA alone, before the more specialized DNA-protein world evolved. This became known as the RNA world hypothesis. But still a fundamental question remained. How did RNA itself first form? RNA is far more complex than the amino acids in Miller's experiment. It consists of long chains of nucleotides, each containing a sugar, ribose, a phosphate group, and one of 4 nitrogenous bases. The spontaneous formation of such complex molecules seemed improbable, especially under the chaotic conditions of early Earth. This is where our story truly begins, with a humble 3-atom molecule that would eventually bridge the gap between geochemistry and biochemistry, between the world of rocks and the realm of life. That molecule is hydrogen cyanide, HCN. Hydrogen cyanide seems an unlikely hero in the drama of life's origins. After all, it is notorious as a deadly poison that blocks cellular respiration. The very name, cyanide, evokes images of murder mysteries and chemical warfare. How could something so toxic be fundamental to life's emergence? To understand this paradox, we must first understand the chemistry of the early Earth. For billion years ago, our planet's atmosphere lacked oxygen but contained nitrogen, carbon dioxide, water vapor, and various other gases released by volcanic activity. Lightning storms and ultraviolet radiation from the unfiltered sun provided abundant energy to drive chemical reactions in this primordial atmosphere. Under these conditions, simple molecules like nitrogen and methane could combine to form hydrogen cyanide. This process has been demonstrated repeatedly in laboratory simulations and is observed today in the upper atmosphere of Titan, Saturn's largest moon, which serves as something of a time capsule of early Earth conditions. Once formed, hydrogen cyanide dissolves readily in water. In the ancient oceans and lakes of Earth, it would have accumulated to form what chemists call a feedstock, a starting material for more complex chemistry. Graham Griffith, a prebiotic chemist at the University of Cambridge, has spent the past decade investigating hydrogen cyanide's role in the origin of life. In his laboratory, housed in a century-old building with modern equipment, Griffith recreates conditions that might have existed in primordial environments. Hydrogen cyanide is remarkably versatile, Griffith explains, adjusting the settings on a specialized mass spectrometer. Under the right conditions, it can react with itself to form more complex nitrogen-containing compounds. Add a bit of iron or other metals common in Earth's crust, and the chemistry becomes even more interesting. One of hydrogen cyanide's most significant reactions is its ability to form nucleobases, the information-carrying components of RNA and DNA. Adenine, one of the 4 bases in both RNA and DNA, can be synthesized directly from 5 hydrogen cyanide molecules. This was first demonstrated in 1961 by Juan Oro, a Spanish-American biochemist, who showed that heating a concentrated solution of ammonium cyanide, which forms from hydrogen cyanide, produces adenine with remarkable efficiency. But the puzzle grew more complex. While adenine could be formed relatively easily, the other nucleobases, guanine, cytosine, and uracil in RNA, thymine in DNA, proved more challenging to synthesize under prebiotic conditions. Additionally, nucleobases alone are not sufficient to create RNA or DNA. They must be attached to sugars and phosphates to form nucleotides, which then chain together to create the full molecules. The sugar component presented a particular challenge. Ribose, the sugar in RNA, is difficult to form selectively in prebiotic conditions. When simple carbon compounds react to form sugars, they typically produce a complex mixture of different sugars, with ribose representing only a small fraction of the total. In 1999, Stanley Miller, the same scientist who conducted the famous spark discharge experiment, along with his colleague Michael Robertson, made an intriguing discovery. They found that ribose could be stabilized in the presence of certain minerals, particularly borates. These minerals, common in arid regions where water evaporates to form mineral deposits, can bind to ribose and prevent its degradation. This finding suggested that life's chemistry might have been concentrated and organized by geological processes, perhaps in shallow pools that underwent cycles of wetting and drying, or in porous rocks where minerals and organic compounds could interact intimately. Another breakthrough came from John Sutherland's team at the Medical Research Council Laboratory of Molecular Biology in Cambridge. Rather than trying to assemble RNA from its individual components, nucleobases, ribose, and phosphates, they discovered an alternative pathway, starting with simple molecules available on the early Earth, including compounds derived from hydrogen cyanide. They showed how RNA nucleotides could form through a sequence of reactions that bypassed the need for preformed ribose and nucleobases. The key insight was that the sugar and base portions of nucleotides could form together, already connected, rather than separately. This dramatically simplified the chemistry required for nucleotide synthesis. And hydrogen cyanide was a crucial starting material in this pathway. But the mystery deepened further. Even if nucleotides could form under prebiotic conditions, how did they join together to create RNA strands? Modern cells use enzymes to stitch nucleotides together, but these enzymes wouldn't have existed before life. In 2013, a team led by Zoe Davidson at the Scripps Research Institute made a surprising discovery. They found that wet-dry cycles, similar to those that might occur at the edge of a pond or tide pool, could drive the formation of RNA-like polymers without enzymes. When a mixture of nucleotides was repeatedly dried and rehydrated, the nucleotides joined together to form chains. It's remarkable how environmental cycling can substitute for biological catalysis, Davidson noted in her paper published in Nature Chemistry. What we're seeing is that geology, simple processes like evaporation and rehydration, can provide the energy needed to form complex biological molecules. These discoveries pointed to a growing realization. The chemistry of life may have been guided by environmental conditions and cycles. Life didn't simply emerge in a static primordial soup, but rather through dynamic processes that concentrated, organized, and energized chemical reactions. As compelling as these laboratory demonstrations were, they still left unanswered questions. Laboratory conditions are carefully controlled, but the early Earth was chaotic and diverse. Where exactly might these reactions have occurred? And how did the transition happen from chemistry to biology, from reactions that happen to reactions that serve a purpose? Scientists began looking more closely at specific environments on the early Earth that might have provided ideal conditions for life's chemistry. 3 settings emerged as particularly promising: hydrothermal vents in the ocean floor, volcanic pools on land, and the porous rocks of geothermal fields. Hydrothermal vents, discovered in 1977, are fissures in the ocean floor where geothermally heated water enriched with minerals flows into the cold, deep sea. These natural chemical factories create steep gradients of temperature, pH, and mineral concentration. Modern vents support rich ecosystems powered not by sunlight but by chemical energy. Could similar environments have nurtured life's first chemistry? Gunter Gustafsson, a marine geochemist at the Woods Hole Oceanographic Institution, has spent decades studying hydrothermal vents. In 2017, his team published evidence that hydrogen cyanide could form in significant quantities in certain types of vents called alkaline hydrothermal vents. These vents are like natural flow reactors, Gustafsson explains. They create tiny compartments in mineral precipitates where chemicals can concentrate and react. The continuous flow brings fresh reagents and removes waste products, not unlike what happens in a living cell. Alkaline hydrothermal vents are particularly intriguing because they produce natural proton gradients similar to those that power all living cells today. Every cell on Earth generates energy by pumping protons across membranes and then harnessing their flow back across the membrane to synthesize ATP, the universal energy currency of life. But other environments have their proponents too. In 2018, a research team studying geothermal fields in New Zealand made a compelling case for land-based origins. These environments, with their porous rocks, fluctuating water levels, and rich mineral chemistry, provide ideal conditions for concentrating organic compounds and driving their reactions through wet-dry cycles. What makes geothermal fields so interesting is their heterogeneity, explains Gabriel Greenwood, a geochemist who led the New Zealand study. Within a small area, you can have extreme differences in temperature, pH, and mineral composition. This creates a natural laboratory where countless chemical experiments run simultaneously. The debate over where life began remains unsettled, but what has become increasingly clear is the central role of hydrogen cyanide in life's chemical origins. Computer simulations of prebiotic chemical networks consistently show that hydrogen cyanide serves as a hub, a molecule from which many paths lead toward biological complexity. In 2020, researchers at the Earth Life Science Institute in Tokyo conducted the most comprehensive computational analysis to date of possible prebiotic chemical networks. They modeled reactions among thousands of plausible prebiotic compounds to identify the most likely pathways to biological molecules. Their results, published in the journal Science, showed that hydrogen cyanide occupied a pivotal position in the network, serving as a gateway to the chemistry of life. But if hydrogen cyanide was so important, what ensured its availability on the early Earth? This question led scientists to look beyond our planet, to the cosmos itself. Studies of comets, asteroids, and interstellar clouds have revealed that hydrogen cyanide is common throughout the universe. The famous comet Hale-Bopp, which passed near Earth in 1997, was found to contain significant amounts of hydrogen cyanide. More recently, the Rosetta mission to Comet 67P/Churyumov-Gerasimenko detected hydrogen cyanide in its icy nucleus. These discoveries suggested an intriguing possibility. Perhaps some of Earth's prebiotic compounds, including hydrogen cyanide, were delivered from space. During the late heavy bombardment period, approximately 4 billion years ago, Earth was subjected to an intense barrage of comets and asteroids. These cosmic impacts could have seeded our planet with organic compounds formed in space. The chemistry of life may have begun in interstellar clouds before Earth even formed, suggests Grace Garrison, an astrobiologist at NASA's Jet Propulsion Laboratory. Simple molecules like hydrogen cyanide form readily in space, where they can undergo further reactions on the surfaces of dust grains. Analysis of meteorites has supported this hypothesis. Carbonaceous chondrites, a type of meteorite containing organic compounds, have been found to contain nucleobases, amino acids, and other biologically relevant molecules. The famous Murchison meteorite, which fell in Australia in 1969, contains over 70 different amino acids, many used by life on Earth. As researchers pieced together the story of hydrogen cyanide's role in life's origins, a more profound question emerged. Was life's emergence chemistry or coincidence? The boundary between complex chemistry and primitive life is not sharply defined. At what point does a system of chemical reactions become alive? To address this question, scientists needed to identify the essential properties that distinguish life from non-life. They needed to understand how a chemical system could begin to exhibit behaviors we associate with living things: replication, metabolism, and evolution. The revolution in our understanding came from recognizing that life is fundamentally an information processing system. What distinguishes living things from inanimate matter is not just complex chemistry but chemistry organized by information. Specifically, hereditary information that can be replicated and that occasionally changes through mutation, allowing for evolution by natural selection. In the modern world, this information is stored primarily in DNA. But DNA is too complex to have formed spontaneously. This is where the RNA world hypothesis gained traction. RNA can both store information, like DNA, and catalyze chemical reactions, like proteins. If the first life used RNA for both functions, it would solve the chicken and egg problem of which came first, genes or enzymes. The turning point in our understanding came in 1982, when Thomas Cech and Sidney Altman independently discovered ribozymes, RNA molecules that function as enzymes. This revolutionary finding, which eventually earned them the Nobel Prize in Chemistry, demonstrated that RNA could indeed perform both informational and catalytic roles. But how did the first RNA molecules form? And how did they begin to replicate themselves? Here, hydrogen cyanide re-enters our story in a dramatic way. In 2009, a team led by Gregory Gendel at the Scripps Research Institute demonstrated a plausible pathway from hydrogen cyanide to RNA nucleotides. Starting with cyanide derivatives and other simple prebiotic molecules, they showed how the building blocks of RNA could form under conditions that likely existed on the early Earth. What's remarkable about this chemistry, Gendel noted at the time, is that it proceeds in water at moderate temperatures and produces the nucleotides with the correct stereochemistry, exactly as they appear in biological RNA. This work resolved one of the most significant obstacles to the RNA world hypothesis: the problem of how RNA nucleotides could have formed prebiotically. But it still didn't explain how these nucleotides could assemble into strands of RNA or how those strands could begin replicating. A major breakthrough came in 2018, when a team at the University of Colorado Boulder, led by Grayson Gilbert, discovered that iron-sulfur minerals, common in both hydrothermal vents and volcanic environments, could catalyze the polymerization of RNA nucleotides. The mineral surfaces not only brought the nucleotides together but also aligned them correctly for bond formation. These minerals act as scaffolds, Gilbert explained. They organize the nucleotides through surface interactions, which greatly increases the probability of polymerization. But perhaps the most stunning revelation came from work on RNA replication. For RNA to serve as the basis for early life, it needed some mechanism of self-replication. Modern cells use elaborate protein machinery to copy their genetic material, but this wouldn't have been available to the first living systems. In 2023, a collaborative effort between laboratories at Harvard and MIT demonstrated something once thought impossible: RNA molecules that can catalyze the replication of other RNAs without protein enzymes. These ribozymes, RNA enzymes, could copy RNA templates with remarkable fidelity, creating complementary strands that preserved the information content of the original. What we've created in the lab is essentially a molecular fossil of the kind of system that could have operated at the origin of life, said Zachary Dalton, the lead author of the study published in Nature. It's not exactly what existed 4 billion years ago, but it shows that RNA-based replication is chemically feasible. This work provided a plausible mechanism for the emergence of self-replicating systems from prebiotic chemistry. The pathway from hydrogen cyanide to replicating RNA now had experimental support at every major step. 1. Formation of nucleobases from hydrogen cyanide. 2. Assembly of complete nucleotides. 3. Polymerization of nucleotides into RNA strands. 4. Replication of RNA through ribozyme-catalyzed synthesis, but the most profound insight came from understanding that this chemical evolution was driven by the same force that drives biological evolution: natural selection. Even before the first cells existed, molecules themselves could undergo a form of selection. RNA molecules that folded into shapes with useful catalytic activities, such as facilitating their own replication, would become more abundant over time. Those that could protect themselves from degradation, perhaps by binding to mineral surfaces or forming simple membrane-like structures, would persist longer in the environment. This concept, known as molecular evolution, bridges the gap between chemistry and biology. It explains how non-living chemical systems could gradually acquire the properties we associate with life. A vivid demonstration of molecular evolution was provided in 2016 by Gerald Gibson's laboratory at Stanford University. They created a system of RNA molecules that could replicate with errors, mutations, and compete for limited resources. Over generations, they observed the RNA molecules evolve greater replicative efficiency, a clear example of Darwinian evolution at the molecular level. What we witnessed was natural selection acting on molecules rather than organisms, Gibson remarked. This suggests that the principles of evolution were operating even before the first cells existed. The picture that emerged was of a gradual transition from geochemistry to biochemistry, with hydrogen cyanide serving as a bridge between these worlds. From this simple three-atom molecule, the chemistry of life could unfold through a series of steps, each building on the complexity of the previous one. But this raised another question. If the chemistry leading to life was relatively straightforward, driven by common molecules and natural processes, why don't we see new life emerging today? Why does all life on Earth share a single ancestry, as evidenced by the universal genetic code and common biochemistry? The answer likely lies in what scientists call the winner-takes-all scenario. Once primitive cellular life emerged, it would rapidly consume the prebiotic molecules that could lead to competing origins. The first successful cells became both the products and the terminators of prebiotic evolution. Additionally, modern Earth has an oxygen-rich atmosphere that would quickly destroy many of the molecules critical to life's origins, including hydrogen cyanide itself. The chemistry that gave rise to life required the reducing oxygen-poor conditions of the early Earth, conditions that no longer exist because life itself transformed our planet's atmosphere by inventing oxygenic photosynthesis. The irony is profound. Life created conditions that would prevent its origin from occurring again. The bridge that hydrogen cyanide once provided between the worlds of geochemistry and biochemistry has been dismantled by the very life it helped create. Yet this realization doesn't diminish the wonder of life's origins. It enhances it. We now understand that life emerged through a sequence of chemical events that, while not inevitable, were favored by the conditions on early Earth. Hydrogen cyanide, a simple molecule formed from the most common elements in the universe, served as a keystone in this process, enabling the chemistry that would eventually lead to the first cells. What makes this story so compelling is that it doesn't require anything mystical or extraterrestrial. The ingredients for life were available on the early Earth, and the processes that transformed these ingredients into living systems operated according to known chemical and physical principles. The miracle, if we can call it that, lies not in supernatural intervention but in the extraordinary emergent properties of matter itself. The realization that life emerged through chemistry rather than miracle has profound implications that extend far beyond the laboratory. It reshapes our understanding of humanity's place in the cosmos and raises new questions about the potential for life elsewhere. First, it suggests that life may be more common in the universe than once thought. If life arose on Earth through relatively straightforward chemistry involving common molecules like hydrogen cyanide, similar processes could have occurred on countless other worlds. Recent astronomical discoveries have confirmed that the basic ingredients, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, sometimes abbreviated as CHNOPS, are abundant throughout the galaxy. The chemistry that led to life on Earth is universal chemistry, explains Gabriela Gomez, an astrobiologist at NASA's Astrobiology Institute. The same physical and chemical laws apply everywhere. If the right conditions exist, liquid water, organic molecules, energy sources, and sufficient time, the chemistry of life could unfold on other worlds just as it did here. This perspective has influenced the search for life beyond Earth. NASA's Mars missions have specifically looked for evidence of organic compounds, including hydrogen cyanide derivatives, in Martian soil. The Cassini mission to Saturn revealed that Titan, with its nitrogen-rich atmosphere and hydrocarbon lakes, hosts active hydrogen cyanide chemistry today, making it a natural laboratory for studying prebiotic processes. Even more tantalizing are the discoveries of subsurface oceans on multiple moons in our solar system, including Europa, orbiting Jupiter, and Enceladus, orbiting Saturn. These oceans, warmed by tidal forces rather than sunlight, could provide ideal environments for life's chemistry to unfold, reminiscent of the hydrothermal vents that may have nurtured life on early Earth. As we extend our gaze to exoplanets orbiting other stars, the implications grow even more profound. We now know that planetary systems are the norm rather than the exception, with most stars hosting planets. Many of these planets orbit within their star's habitable zone, where liquid water could exist on the surface. If the chemistry of life is as robust as our research suggests, living worlds may be common in our galaxy. But the implications extend beyond astrobiology to philosophy and our understanding of human identity. The chemical origin of life reveals that we are intimately connected to the cosmos in ways ancient philosophers could only intuit. The atoms in our bodies, the carbon in our DNA, the nitrogen in our proteins, the oxygen we breathe, were forged in the hearts of stars that exploded billions of years ago. Hydrogen cyanide, the molecule that helped bridge the gap between geochemistry and biochemistry, forms readily in interstellar clouds. The chemistry that led to us began long before Earth existed. We are, in a very real sense, made of stardust, assembled through processes that began in the vastness of space and continued on the surface of our young planet. This cosmic perspective challenges traditional notions of human exceptionalism. We are not separate from nature but embedded within it, the product of the same chemical and physical processes that operate throughout the universe. Life is not something imposed on matter from outside but rather an emergent property of matter itself, given the right conditions. Yet this naturalistic account of our origins doesn't diminish human significance. It enhances it. Understanding the improbable sequence of events that led to our existence, from the formation of hydrogen cyanide in the primordial atmosphere to the emergence of self-replicating RNA, from the first primitive cells to the complex organisms we are today, can inspire a profound appreciation for the preciousness of life. When you realize how many things had to go right for us to be here, reflects Grayson Gilbert. It fosters a sense of gratitude and responsibility. We're the products of 4 billion years of cosmic and biological evolution, the universe becoming conscious of itself. This perspective also has practical implications for how we approach environmental challenges. Recognizing that our biology is chemically connected to Earth's geochemistry underscores our dependence on planetary systems. The elements cycling through our bodies today, carbon, nitrogen, oxygen, are the same elements that cycled through primordial oceans and atmospheres. By disrupting these biogeochemical cycles through pollution and habitat destruction, we ultimately harm ourselves. Perhaps most significantly, understanding life's chemical origins helps dissolve the perceived boundary between the living and non-living worlds. Life didn't appear suddenly as something categorically different from its surroundings but emerged gradually through increasingly complex chemical systems. The transition from chemistry to biology was not a moment but a process, one that unfolded over millions of years as molecules gained the ability to replicate, metabolize, and evolve. This continuity between the living and non-living has implications for fields ranging from artificial intelligence to bioethics. If life emerged through the self-organization of chemical systems, could we create new forms of life in the laboratory? Scientists are already synthesizing genomes and creating minimal cells. The ethical questions surrounding such work become more nuanced when we recognize that the boundary between design systems and natural life is not as clear-cut as once believed. Similarly, as artificial intelligence systems grow more sophisticated, questions about consciousness and sentience take on new dimensions. If consciousness emerged in biological systems through natural processes, could it also emerge in sufficiently complex artificial systems? The chemistry of life's origins suggests that remarkable properties can emerge from systems organized in particular ways. For now, these questions remain speculative. But the demonstrated pathway from hydrogen cyanide to the chemistry of life provides a solid foundation for further exploration. It reminds us that the most profound transformations can begin with the simplest of ingredients, given sufficient time and the right conditions. As we continue to investigate the chemical origins of life, both in the laboratory and on other worlds, we are engaged in one of humanity's most profound intellectual adventures. We are uncovering not just how life began but who we are at the most fundamental level. Temporary assemblages of atoms with the remarkable ability to contemplate our own origins. In the end, the story of hydrogen cyanide and life's emergence reveals something both humbling and exalting. We often think of life as fragile, and individual organisms certainly are, but the chemistry that produces life may be robust and common. Given the right ingredients and conditions, the universe seems predisposed to create complexity from simplicity, organization from randomness, and eventually consciousness from chemistry. The cosmos is not merely a collection of dead matter punctuated by rare oases of life. Rather, it contains within its basic laws and elements the potential for life. Hydrogen cyanide, a molecule so simple it consists of just 3 atoms, serves as a bridge between worlds seemingly separate: the geochemical and the biochemical, the inanimate and the alive. When we look at a rock today, we no longer see something fundamentally different from ourselves but rather a distant cousin, matter that followed a different evolutionary path. The atoms in that rock could, under different circumstances and through hydrogen cyanide chemistry, have become part of a living system. This perspective doesn't reduce life to merely chemistry. It elevates chemistry to something capable of producing the wonder of life. The gulf between rocks and living cells, while vast, is not unbridgeable. It was crossed once on Earth through a sequence of chemical reactions, with hydrogen cyanide serving as a crucial intermediate. As we ponder our place in the cosmos, we can find profound meaning in this chemical connection to the universe. We are not accidental tourists in a foreign reality but native inhabitants, expressions of the creative potential inherent in matter itself. The hydrogen cyanide that helped spark life on Earth forms naturally throughout the cosmos, suggesting that the universe may be filled with experiments in complexity, consciousness, and life. The most awe-inspiring aspect of this scientific narrative is that it reveals a universe more extraordinary than any creation myth. A universe where planets, stars, and perhaps even life itself are not exceptional anomalies but natural expressions of physical law. We exist because the chemistry of the cosmos allows for us, perhaps even encourages us. From the simplicity of hydrogen cyanide to the complexity of a conscious human being contemplating the stars, the universe has found a way to know itself through us, its atoms arranged into systems capable of discovering their own cosmic origins.